Hey guys, my name is Carter, and we're going to be talking about sensory neurons, specifically low threshold mechanoreceptors. It's a little long, so I have to talk a little bit quickly, so sorry. Sensory cells, just a basic overview from the book, are simple cells that link skin and muscles to the spinal cord. They are a T-shaped structure, like in the diagram in the bottom right corner. The cell body has no dendrites and plays no role in shaping how information is transmitted. The communication was originally discovered by Edgar Adrian when discovering muscle stretch. And then different sensory cells can give different kinds of information about what is happening on our bodies or on our skin. And there are several different kinds of sensory cells. There are three types of somatosensory system functions: exteroceptive, which is perception and reaction to information outside of the body; interoceptive, which is perception and reaction to information inside of the body; and then proprioceptive, which is perception and control of body positions and balance, much like your awareness of where you are within the world. There are mechanosensitive sensory neuron types. There can be broken down into two kinds: low threshold mechanoreceptors, LTMRs, which are innocuous mechanical stimulations. And then high threshold mechanoreceptors, HTMRs, are harmful or potentially harmful mechanical stimulations. Here is a fun picture of all of it in work that will make sense after if it doesn't now. There are slow adapting and rapidly adapting mechanoreceptors. Slow adapting continue to respond to the stimulus after it is removed, and it is better at responding to constant stimulation or sustained indentation. There are two types of slow adapting LTMRs, slow adapting 1 and slow adapting 2. Within rapidly adapting, it is better at responding to movement and like quick stimulus applied. It does not continue responding after the original stimulus. It is better at detecting changes, and there are two types of rapidly adapting low threshold mechanoreceptors: RA1 and RA2. First, SA1 low threshold mechanoreceptors have a conduction velocity of A beta 16 to 96 meters per second. And it is found in both glambrous, which is non-hairy, and hairy skin. In glambrous skin, a cluster of Merkel cells in the basal layer of the epidermis sends signals to one slow-adapting low-threshold mechanoreceptor. In hairy skin, clusters of Merkel cells are also at play surrounding guard hair follicles like in the picture. These mechanoreceptors can transmit a highly acute image of tactical stimuli. They are mostly activated by indenting the skin, by poke or hitting a corner or something like that. Whether the Merkel cell or the A beta SA1 or both are sites of initiation of the slow adapting low threshold mechanoreceptor responses, remains a topic of considerable debate. Next are slow adapting 2 low threshold mechanoreceptors with a conduction velocity of A beta 20 to 100 meters per second. They are also found in both glambrous and hairy skin. In glambrous skin, the responses possibly stem from the Ruffini corpuscles in the dermis. They have been identified in hairy skin, but are largely unknown and remain controversial. They're also sensitive to skin indentation, but are much more sensitive to skin stretch and changes in hand fingers and shape, similar to proprioception. Also sensitive to object motion and velocity in relation to the skin stretch. The Ruffy ending is unconfirmed. That is why there's a question mark in this picture. Next are rapidly adapting low threshold mechanoreceptors. They have a conduction velocity of A beta 20 to 100 meters per second, like slow adapting. They are also found in glambrous and hairy skin. In glambrous skin, Myciner corpuscles are associated with them in the dermal papillae. And in hairy skin, responses arrive from longitudinal lanceolate endings surrounding the guard and or all on achene hair follicles. They are sensitive to object movement, hair follicle deflection, and low frequency vibrations. Um, RA and SA LTMRs work together similarly to the roles of rods and cones, which I thought was a cool fact. Next is RA2, LTMRs. They have a conduction velocity of a beta 30 to 90 meters per second. They are only found in glandulous skin and are correlated with Pacinian corpuscles in the deep dermis. They are largest density in fingers, but also found in joints. They are sensitive to high frequency vibrations in the hand and respond to amplitude thresholds lower than rapidly adapting one. The receptive field sometimes encompasses the entire hand, making it unable to detect spatial acuity. There are other low threshold mechanoreceptors that I did not discuss in this presentation for lack of time, and they work together with slow adapting and rapidly adapting to play an important role in touch perception. So that's kind of represented in this picture here. Here are my sources. Thank you so much.